So our first speaker for today's opening plenary session is the Senior Statistical Advisor for the Office of Translational Science and the Center for Drug Evaluation Research, CDER, at the Food and Drug Administration. Until June 2011, Dr. ONeil was the Director of the Office of Biostatistics at CDER, which provided biostatistical and scientific computational leadership and support to all programs. Prior to October of 1998, he was Director of the Office of Epidemiology and Biostatistics, responsible also for the post-market safety surveillance of new drugs. Dr. ONeil was the ICH FDA topics leader for two guidances, E6 and E5. He is a fellow of the American Statistical Association and the Society for Clinical Trials. Please welcome Dr. Robert ONeil.
Thank you, Susanna. And thank you for inviting me here. Let me see if I got my act together here. Make sure I can advance these slides. Eric, you may need to help me here with this. That's okay. I got it. All right. Thank you for inviting me here. I've never talked in front of this society before, but I'm not going to restate the title of my talk, but the theme here is the long view. And this is a traditional FDA disclaimer. But the motivation for this talk today came from a meeting that I had at Harvard with Eric, and he invited me to give this talk. And I said, well, what can I say that might be of interest to this crowd? And that meeting at Harvard was on competencies in clinical research. And so I started to think about this, and I've been involved in a lot of this in some ICH GCP training and for international regulators and whatnot. So this is what I put together. This is motivation for my talk. Primarily, as you heard in the introduction, you're a fairly diverse population. In my mind, that comes with a lot of pros and cons because it's a big complex and you have to know what your buddy is doing. So understanding your role in contribution relative to what your other colleagues are doing to the quality and relative to others on the team. And that's the long view. What happens after this trial is completed? And do you know the process that's going to follow, how the data is cleaned up, where it goes, when it's locked, when it's analyzed? And at the end of the day, if this is a product that is going to go on the market, it'll go to the FDA. And this is what happens when it's under review, and understanding what goes on when it's under review. So this is, the motivation is for how it all fits together. So some of you are probably aware that there's a lot of things going on in the clinical trial paradigm. They're becoming more complex. The management of them is hard. The cost of them is hard. There's a lot of interest in real-world data and how one might move into that world in doing clinical trials in real-world data, use of electronic healthcare records and whatnot. So the whole skill mix that's needed in this game is starting to change. And how might you think about this in terms of accommodating it? And how do we train people for what's happening in the future to maximize their contributions? And so finally, at the end of the day, part of the rationale for why you're here is understanding how the entire clinical process works, how it fits together, what your role is, and how you affect it downstream. So when you look at this, many disciplines look at it through their lens, but you really have to think about the long view, in my mind, in terms of whose lens you're looking through, and all the lenses, if you will. Some of you are aware probably that ICH-E6, the GCPs, became revised and finalized in November of last year. And it was revised for a reason, because of the increasing complexity and in some ways naivete of the original ICH document, which was written 20 some odd years ago, and the cost of clinical trials. And it never really recognized the advances in electronic data capture and everything else that's going on with the recording of outcomes and mobile devices and all this other stuff that's going on today. The original GCPs never envisioned that. And then finally, there was a whole culture change in terms of not a checklist, but thinking in terms of the long view, scenario planning, quality by design, thinking about it in advance. How is it going to play out? How many ways can it go south? And so there's a lot of emphasis on the clinical trial complex, everybody in the game thinking about how it may play out in the future. And then the last piece, which is central monitoring. There's been a lot of on-the-boots site evaluation, cleaning up of data and whatnot, but there's also interest in central monitoring, and I'll return to that in terms of how that might work. So one of the points here is that GCP training is important, but it's not sufficient for the team sport that you're in. And GCP training in clinical trials and just the conduct of clinical trials is very important. But your understanding of the context in this and how it, at the end of the day, impacts public health. I think you've heard a couple of comments about how important clinical research and clinical trials are until your actual personal experience. Probably everybody here has had some medical treatment or drug treatment that has been impacted by good quality clinical research that set the standard for how you're going to be treated. So one of the, I don't know how many of you are involved in multi-center studies, but most clinical research is a multi-center study these days. And in fact, most clinical research that comes to FDA is 60% to 70% outside the United States. So essentially, these are trials that are done in multiple regions. They're done in the States, North America, Latin America, Asia. And so think about the design of that trial. Think about everybody who is part of the game to assure comparability that the protocol is being carried out the same in all these different regions that might have different medical milieu and whatnot. Just think about the training of that enterprise. So, and I've been involved a lot in that. I've published a fair amount about it. And I was the ICH topic leader in E6, which was the acceptance of foreign clinical data. So it's a really important issue today. This article here in the recent DIA journal is worth a read. It's sort of the theme on GCP training is only one part of the preparation of what you do in conducting a clinical trial. Protocol-specific training and opportunities to gain experience in clinical research through mentorship and other avenues is very important. So the theme here is the downstream impact of your efforts to support the approval of a medical product. Not all trials are done for that purpose, but my particular focus and my interest right now is on this for this meeting. Competence is not a checklist. It's a sort of a thought process. It's a culture, if you will. The protocol is as important as complying with the GCP, and I don't know to what extent you go through the protocol before you or companion with training with the GCPs, but that's a really important part of this whole process. And at the end of the day, if these trials come to FDA, I'm going to walk you through what some of the FDA does in terms of looking at the data that you produce. And so the quality of the data is critical to whether that trial is a success or a failure. And that's part of what I wanted to get across in the theme of this particular talk. So, as I said, this is a team sport, and everybody's working towards a similar goal, and that's the success and the interpretability and access by patients to the therapy and the product that you're evaluating. So let's talk about the protocol. It's almost common sense that you want to make sure that patients are eligible for the trial and meet the protocol-defined eligibility. And does the subject have the disease properly classified according to the protocol? And this may not be as easy as it seems. This may be, depending upon the stage of the disease, depending upon the kind of biomarkers that are used now to qualify you for the trial. That's not always that easy. And in targeted therapy trials, this is a big-time issue. Because if you're in an enrichment design, which essentially enriches for targeted therapy for breast cancer, and you don't have that target, this product isn't going to work for you. So you're going to downweight the treatment efficacy because you're entering subjects, even though you randomize them, that are not going to respond to therapy. So that's a big ticket item in personalized medicine and targeted therapy. And are you clear on what the primary endpoint is and how it's adjudicated? And you better make sure usually that it's adjudicated in a blind manner. If it's not, you can have differential outcomes solely because people know which treatment you're on. And the other aspect, which is receiving an awful lot of attention, both nationally and internationally, is the issue of follow-up of patients. If you randomize a patient into a trial, you cannot do an intent to treat analysis of everyone randomized, analyzed, if you don't have an outcome for them. So if they leave early and you don't have an outcome for them, that's a problem. And if you have too many of them, it's a real problem because the trial becomes uninterpretable. And if it actually is differential in terms of withdrawals on one treatment versus another, it becomes that much more uninterpretable. And I'll show you a couple of examples a little later on. So this is on the protocol side. You may be aware of these initiatives. There are two initiatives for common protocol template to improve the efficiency and consistency of clinical trials. One is coming out of the NIH FDA effort, and NIH essentially says, if we're funding you for an NIH trial and you're doing a trial under an IND, you have to follow this particular template. And they've created an electronic template which makes it easier for the whole team and the clinical investigator to write the protocol. There's a similar effort going on in the industry from the Transellerate crowd, which is a nonprofit industry sponsored, that is paralleling that, so that all the industry would not have to do one-off reinvent the wheel for templates for clinical trials. So these are two areas that you should probably be aware of, and at the end of the day should be part of your training, or should be moved into GCP training, in my mind. Some protocols are becoming very complex and requiring a lot of planning. The big ones, the large outcome trials, the diabetes trials, the 10,000 patient cardiovascular trials, those are big, tough trials to do. But there's a lot of history on their outcomes and adjudication of their outcomes and whatnot. But the other thing that's going on is site variability. So when you have a 300-site clinical trial that's done all over the world, who's looking at the variability? How much is too much? How much is bias? How much is real? How much is interfering with the assessment of the treatment effect? Is the treatment effect consistent throughout? Does it apply to everybody? So one of the big issues in ICHE 6, acceptance foreign clinical data, is if the same trial is being submitted to Europe, the United States, Japan, and other Asian regions, does everybody get the same message out of that trial in terms of whether it's going to support the efficacy of a treatment in their region? So that's the other region, the big ticket item with regard to assessing variability and consistency of treatment effect throughout sites, not just sites, but primarily regions within sites within regions. And now there's a whole new area that's coming about with regard to what's called master protocols. And they're going on in the lung cancer area, they're going on in the breast cancer area, where you have multiple companies that are putting their product into the same trial with a common control group. And then essentially nobody's being compared with each other. They're just being compared with a common control group. And then there's another one called the platform trial, which essentially is if you have a cancer that is targeted by a genotype, but it's not necessarily breast cancer and lung cancer and skin cancer, and different cancers all have the same genetic fingerprint, then all those cancers are being evaluated in the same trial. So and then there's the whole issue of adaptive trials, which are trials that are modifying themselves during the conduct of the trial, conditional on what you're observing during the course of the trial. So all this stuff is going on. So and I'm not even talking about orphan and rare diseases. That's a whole other conversation. Those pediatric and orphan rare diseases, they're very difficult issues. The designs are somewhat different than in the more traditional areas, and I'm not even talking about that. So here's just a visual on the case report form relationship to the protocol. The case report form in the old days, certainly in the old FDA days, would be essentially 3 to 4 inches of paper coming in, which would have everything but your shoe size that would be on those forms. But when you boil that all down to what is being analyzed and what's the critical stuff, you essentially separate that out into how much of it is going to be part of the protocol that is going to be analyzed, how much of it is going to be the safety profile that is going to be analyzed, and what is it important about the notes versus the outcomes that are being assessed, that are being analyzed. So visually, you can think about the case report form being segmented into these 3 areas. Now, right now there is a requirement with data standards that's coming about, I think momentarily. It's already in force. And I'm not going to spend any time. You can go to the FDA website on this. But there's more than bedtime reading here for you to understand what the importance of data standards in the format. Because if you don't collect it that way, it's not going to be acceptable, and it'll be dumped back to you. So there's a requirement for data standards, and there's a website that walks you through a lot of this. And these are resources available to you. I'm just hoping this guy, yeah. Then there's this other data standards on CDISC, some of this Clinical Data Interchange Consortium. Essentially, FDA is requiring this as this year for all electronic submitted data. So again, there's a web page for this crowd. I'm not going to go through it in any detail, but CDISC standards specify how to structure the data. They do not specify what data should be collected or how to conduct clinical trials, assessments, or endpoints. It's just a matter of the standards for that data. And this is important so that as we get into the world of meta-analyses and looking at trying to assess what went wrong with these clinical trials, your ability to be able to go in and look at 10 different diabetes trials, 10 different heart failure trials, 10 different antiviral trials, and say, what was good about these trials and what was bad about these trials? You can't analyze these across different trials if you don't have common data standards. So in the old days, you had to recondition all the data to do that kind of analysis. And that's just unacceptable in 2017. You just can't do that. You don't have time, it's inefficient, and it's not a good way to reuse data. So as you get into all this area of sharing data, you have to have standards. Otherwise, sharing data is useless. So anyway, that's the point behind this. Then within the data model, there's two issues here. One is the study data tabulation model. This essentially is how do you structure the individual patient profile, all the data that you're collecting on an individual, how do you structure that data? And that's what the study data tabulation model is. And again, you can go to the CDISC website and get schooled up on this. Then there's the analysis data model. So among all that data that's collected on the individual, there may be 20 data points that are used for the analysis, the survival analysis, the longitudinal analysis, the analysis on repeated visits, covariate analysis, all that kind of stuff. That's coming out of the analysis data model, and there's standards for that. So that's that part. So at the end of the day, you may not be involved in this, but you may be part of this. You're contributing to the data collection, but there's good clinical practice guidance. There's data management that's going to have to happen after it's out of your hands. There's the data storage, data usage, and the data auditing, site selection, and someone may eventually come back and do a site audit on that data. And what are they going to look for? So what happens with this clinical data when it comes to the FDA? So this is usually done not by an investigator, but it's done by a sponsor for whom you might be doing the trial. So let's say a Pfizer or a GSK or a Merck or somebody, they're sending this data into the FDA. It gets a quick look to see whether it's reviewable from an electronic data standards and format perspective. If not, it goes back. It passes that, and then it gets a scientific review and analysis, possibly a reanalysis of the data, conduct, potential biases, quality, strength of evidence for a benefit-risk decision. Then there may be a public advisory committee. If we have time, I'll have a 4 or 5 minutes clip on this. And then some trials and some sites are going to get audited for every new drug application. And there is a site selection tool that FDA uses. So if you look at the time frame of this, essentially here's a 10-month time frame for when a sponsor decides to send this into FDA. The FDA has a set time to be able to review and come to a decision on this. So the quality of that data, the ease of access to that data, and all this stuff is very important to the efficiency of the process. So essentially, by the end of the first month, FDA is planning its review of the data that's been submitted by a sponsor. And then by month 9 or 10, there's an official action that's taken. So primarily, there's a number of different disciplines that are reviewing this application. The medical clinical review and the statistical reviews of efficacy and safety are the ones that I'm most familiar with. And this is the clinical folks and the statistical folks working very hand in hand, close together as a team. And the clinical study report that is submitted is in ICH E3 format. It's all the electronic data on the patient level. That's the patient level data tabulations, case report form. That comes in. The protocol and all the amendments, any changes, what was planned, what was changed. the analysis data set for efficacy, and the safety data set. As I said, the data tabulations are essentially patient-level profiles. The patient-level safety data is sort of separated out and looked at, and I'll go through and give you some idea of how that's looked at. And the analysis data set is really what the statisticians spend most of their time on. That's to confirm the analysis that the endpoint has been met and whether treatment effect has been demonstrated beyond chance. So the statistician spends a lot of time on what was planned, what was the hypotheses. There was a time where, and there still is in a lot of the medical literature, where you can't determine what was planned and what was decided on after the fact. Hopefully those days are over. But the planned hypothesis, what are the primary and secondary endpoints, and was the randomization scheme adhered to in a good one? It's very important. What was pre-stratified, what was the pre-specified analysis plan, not something that you thought about after you had the data in hand. So there's a huge distinction in the regulatory setting between exploration and confirmation. So, and often a clinical write-up may not distinguish that. And what you see in the medical literature may not be what is submitted to the FDA. And I'll spend a little time on that. So the analysis of the patient-level data involves pre-treatment measures, all covariates, baseline pre-treatment measurements that might be relevant to evaluating change or change from baseline or progression or whatever. And then all the treatment measures and outcomes, study features such as patient withdrawals. That's a big ticket item, patient withdrawals. So the evaluation of bias in the conduct of the study, the analysis or the interpretation, and the uncertainty of the result in decision-making. Statisticians spend a lot of time on quantifying the uncertainty of that result. And is that result due to the treatment, or is it due to some bias in the conduct of the trial? That's where you guys come in, because you are essentially conducting the trial, introducing the quality into the trial, maintaining the quality of the trial. And so, what do I have to?
Oh, yeah,
I have till 10 o'clock. Okay. All right, so the law requires that a clinical trial to support an approval be supported by adequate and well-controlled clinical trials. That's generally a randomized trial, blinded, not often, not always, either a placebo or an active control trial, randomization, and all the stuff that you know. But I'm not going to get into the protocol because there are many different protocol designs, many different clinical trial designs. A statistically demonstrated treatment effect, that's where the size of the trial comes into account, and whether you want to get a claim for secondary endpoints. There may be multiple secondary endpoints. Whether the hypotheses tested are the primary endpoints, they should be. If they're not, somebody didn't pay attention to the protocol in the first place. And you know, a lot of protocols are evaluated by FDA in advance, so they're given. The FDA gives sponsors some advice, and the sponsor can take it or leave it. But and then finally is trial bias from the design, from its conduct or its analysis responsible for the result, and obviously data quality and validity. Here are some tools that are used to evaluate data quality and validity. The clinical investigator site selection tool, which eventually is not publicly available but is used by FDA. You guys are probably aware of it because sponsors are requested under a guidance, which I will mention, about what is desired to be submitted to FDA so they can do an application of the site selection tool. And then J review is what primarily the clinical reviewers, the medical officers, used to evaluate the safety of a product. And this is really using access to patient-level data. And this is just a private, publicly available clue points. FDA has a commercial, CRADA a research effort with this company. But there are, I'm sure, other people doing this. But it's statistical central site monitoring. This is just an example from the use of J-Review, which what this is everything that happened in an individual. This is a one-patient profile. And you can't read it, but if you go on the website, you could probably get a little more background text on it. But what's on the top is every concomitant medication that individual's taken from when they entered into the trial, what were the assigned treatments and how long they took them, what were any of the lab values that they got, what are the outcomes that were measured, which are maybe the primary outcomes. So you get a chronologically time-dependent picture of what's happening to this patient. This patient then is wrapped up with all the other patients. A medical officer might be able to see, well, geez, this individual had an adverse reaction at 4 months. What happened preceding that? What treatments were they given? What drugs were they given? Was it a concomitant medication interaction? And did they drop out of the trial? So this is more from the website on J-Review, which you can go to. It's out there, and it shows you the different capabilities of what these programs can do. Some of them just do graphical profiles. Some of them can do patient subsetting. They can look at, they can condition, say, give me all patients with X, Y, or Z, all patients who entered this way, all patients who entered that way, who had this happen to them. And you can get these kinds of plots, these graphical plots. Some can be time-dependent, Kaplan-Meier type of curves, time-to-event kind of curves. The heat map essentially could be age by some baseline value, and the redder the area is, the more stuff is going on, so that's where you might want to look. So essentially, the clinical trial data is evaluated by using some of these tools that allow you to look at what's going on at the patient group level and the patient individual level. The central statistical monitoring piece of this is essentially a sponsor is using this to look at the 300 sites that they have. How are they varying relative to each other? Who's the outlier? Who has more adverse reactions than the other? Are they out of balance or something is different? So you're using this primarily for outlier analysis and consistency and homogeneity analysis. So this is a guidance which is asking sponsors to submit data to FDA, and they submit data to FDA, and this again is the time frame from when it gets submitted, and then the site selection tool is started to be looked at very early in the process. So before the time is up for FDA to make the decision, they have to say which sites are going to go out and be inspected. So how do they do that? In the old days, it was pretty hard. Today, it's getting to be more systematic in terms of it's data-based, and it's looked at where are the sites that are contributing the most inconsistency, maybe consisting the most dropouts, things of that nature. So what you're looking at here is using the clinical investigator site selection tool for data irregularities, outliers, regional and country-specific differences, comparison of variables across treatment arms, things of that nature. And this just gives you another a graphic of what this software looks like to a reviewer who's using that. What you're seeing across the thing are maybe different sites. And you can pick a variable and you can look at what the mean, let's say if it was blood pressure, whether it's a cholesterol level, whether it was this or that, whatever the variables are, you can look at them across sites to see what's going on relative to one site versus another. So that's how variability is looked at, because you have to analyze this data at the end of the day. So the statistical component of this is the variability and the consistency. This is just another graphic that helps one look at where are the sites that might be more outliers or irregular in terms of the data that's being collected. And you can actually think about this being evaluated in process. So if you got a 2-year-long trial, you ought to be looking at this, month by month by month, not necessarily unblinded by treatment, but certainly you can get an idea of, and whoever might be the CRO, who might have access to this kind of stuff, and they're doing central monitoring also, you ought to be in communication with them. So anyway, this is just another graphic, and just to show you that there are tools to be able to look at data irregularities and then do risk ranking of sites where you might want to go to inspect them. Here's just some stuff on why do studies fail, and where do you find information on this? Well, it's not always that easy to do. I'm just going to provide you with some selected articles on a topic that are published by FDA scientists. And you can also use the FDA Advisory Committee transcripts, which are in the public domain. So if you wanted to go to FDA's Advisory Committee public websites, you can get a lot of information there about what happened. I'm going to use a video clip if I have time. So this is just a restatement of what FDA gets. FDA gets everything that is not in the journal article. The journal article might be 5 to 6 to 7 pages, and it can only do so much. So the full study report that FDA gets tells you a lot more, along with the raw data, and that is not necessarily in a published article. So a published article might read one way, and FDA might have a totally different view of that, depending upon how much was described in the report and how much it held to the original protocol and whatnot. This is just an article in the British Medical Journal a year and a half or so ago that compares what comes about in the public domain and the letters that the FDA sends to a sponsor saying, go do another study, or this is what we didn't like about the study that you did. And some sponsors don't share that with everybody. There's a big interest in making this mandatorily publicly available. And this tells you pretty much about what the complete response is that an FDA would send to a sponsor. Here's a publication by FDA scientists. They went through 12 years worth of new drug applications, and essentially this breaks down which ones were approved and were they not approved, and why were they not approved for various reasons. And again, you have these slides, so I'm not going to go through them in any detail. You have the slides, you can get the references and read the articles if you're really interested. But what I outlined in red here are the probably 10% of those that were not deficient were deficient was because the population was not appropriate to reflect the treatment effect. So, you would think you could fix that. And in another case, the endpoints were unsatisfactory in about 18% of the case. You would think you could fix that at the design stage. Another one where the study outcomes were, I think, multiple endpoints, but they were not adjusted. So people said, well, I'm looking at these 5 things, but they didn't adjust for them statistically. There's another article here about, in the British Medical Journal, on making the same point regarding the full clinical study report submitted to the FDA does not provide the same information that is in the published in the published literature. I am going to use a video clip now because I think it's instructive. And so just before you put this on, let me just go back and give an intro on this. This video is of FDA has 17 advisory committees, and most new drug applications that have important new molecular entities come before an advisory committee. An advisory committee is publicly held meeting, usually has 10 to 15 outside independent FDA advisors, usually from the academic world, NIH world, and they advise FDA on the evidence, and it's advisory. And these are now, if they held in FDA's great room on the campus, they're videotaped. So I've been using them at a course at Harvard School of Public Health, essentially as poster children for, here's what happens on a tough call, okay? And you guys can listen in on this stuff too. Just go to any of these that are webcast. But this is January 2012 Reproductive Health Advisory Committee, and it was on a progesterone gel for preterm birth prevention, for preterm birth use, essentially to minimize preterm births. And the issue was 28% non-compliance, and the results in the U.S. and U.S. were not the same. So the chair was, they had already made the vote. These are long meetings, all-day meetings, so I can only do the one clip. So if you can put this guy on. It's not TV quality video, but it is what it is.
Do you believe that there is any clinical explanation based on the data in the NDA for the difference in efficacy results in the U.S. and foreign populations? If yes, do you believe that the explanation could be adequately addressed in labeling so that the progesterone gel could be safely and effectively used in the U.S. population? So, thoughts about the differences between the U.S. and non-U.S. Populations in the study, and then whether or not this could be explained in a labeling from the FDA. So I'll open it up. Dr. Green?
Yeah, I'll continue my harangue from when I explained my vote, which is, as Dr. Darney showed in the subgroup analyses, he mentioned several new drugs that had been approved by the agency recently, where they addressed the issue of subgroups in whom the drug appeared not to work, and they addressed that in the label. It seemed not to work very well in the U.S. population, where noncompliance was at its highest, and I would suggest that the label could address that by saying that the drug won't work if you don't use it.
Dr. Davidson?
I really don't want to be heard. Now that I've passed that truth, I don't think there is a satisfactory explanation for this, and whatever is suggested is purely speculative.
Dr. Emerson?
I'll just expand on what Dr. Johnson said in her explanation before, and that is that at the level of taking the history of women who've had early delivery before, this apparently was a very small subgroup. Now, recognizing that the cerclage is an open question, it's still that it's a very small subgroup if that it couldn't show through as an effect. And again, it's just the the heterogeneity of the disease is such that there's undoubtedly some women who are prone to early delivery, and the shortening of the cervix is caused by that, and that perhaps there's other women who the shortening of the cervix is perhaps one of the earliest signs of the disease. And we certainly don't have any sort of mechanisms being put forward to give us an idea as to which is which in this situation. And I think that further studies could pick this out better than us just leaping to conclusions in a current indication.
I'd just like to make a comment that I did find it somewhat concerning that just 7% of the participants, if they are removed, that it was no longer significant. So I certainly would say to the sponsor, as they prepare to do a further trial with this medication, to just look at those sites and assure that there was no potential downside to those sites. And I think it's, even though I would agree with Dr. Davidson, I do not think that we can find an explanation for this difference, that certainly engaging more of the U.S. population would be reasonable in further study.
Thank you. Along the same line, the issue seems to be the U.S. population. Would it be appropriate? It seems that the question we're begging is that if another study is done, it should be done exclusively in the United States, and it should be powered by a large enough sample size to answer the question, and that if the sponsor elects to do that, then the communication with the FDA needs to be better than it apparently was this time around, because what I'm hearing is, okay, we've agreed to do this, and the data's presented, and the analysis is, no, that's not what we want to look at, regardless of what you did and what we agreed to earlier. And I think that's a real communication problem that has left us with more questions than answers here.
Okay,
Dr. Henderson.
you can probably cut it
I would
now.
like to add to Dr. Harris's point.
Can you-
One of the
can you
things,
stop
something
this?
very simple, basic kind of social science stuff.
All right. Okay, thanks a lot. So I think you get, there would have been one more woman here. What you've been hearing are all the advisory committee members giving their reasons for why they voted the way they did. This was a 14-0 down. Didn't vote for the approval of this. I just wanted to give you a sense for the importance of the site selection stuff, because the next woman would have said, let's go and do some more in-depth evaluation of the sites, and I don't think that's, everybody said that it's all speculative. So this is my recap. What I tried to do here is to go through the competence issue and say it's a big long-term view. It's a lifetime learning effort, you know, and quality by the design is really a new paradigm. Understanding the long view and the process is very important. And most clinical trial research, if it's going to have impact on the public health, probably will come to FDA for review. So I tried to give you some sense of what happens when it does come to FDA and the level of depth that goes down to look at the quality of the data and the interpretability of the study. So thanks a lot.